So it's my pleasure today to be introducing soon-to-be master Daphne Shen. So as many of you guys know, before I start where I'm evaluating whether I'm going to take a graduate student, I do a little bit of an evaluation where I kind of look at social media and try to track down pictures and evidence of what they're really like as a potential student. Some of the things I evaluate are things like, do the students love animals? And when I was looking for different pictures, there seemed to be quite a bit of evidence that Daphne was a fan of animals. She seemed to like little turtles, medium-sized turtles, and large turtles. I got a little concerned at one point when it looked like she might be running off with the animals. So was there a potential that she loved animals a little too much? So when I kept looking, I realized, well, that was all in order to set them free. So one of her goals there was to increase hatchling success. And in addition, I found out that she had an additional skill that would be very useful in our lab, which was gluing things to animals. So she does seem to like turtles at least, but does she like other animals, like mammals, since that's a lot of what we do in this lab or a lot of what we work with? And it does look like she is, a big fan of all types of animals. There she is with a little bunny. This is heading in the right direction. Some beluga whales in the background. And she even seems to like the little rodents there. You can see in the little corner. And they also seem to love her. So from this kind of first look, it looks like she does love animals. And there was this added skill that she knows how to glue things to animals. She can attach tags. Does she have any evidence of being able to kind of charm or tame the animals she works with? There was some evidence that she plays the flute, so maybe she can use that. Then again, that mouse seemed to be able to be tamed by her, and it even seemed to pass on to seals. So that was looking in her favor. And then one of the things that we use a lot when we're working with elephant seals is we have a large red tarp that we use to move animals. And so I was pleasantly surprised to find that she had experience kind of maneuvering large red tarp-like material. So that could become quite handy with seal moving. So I was very excited when she decided to join the lab, and we got her plugged into one of the elephant seal projects. She quickly became an essential team member. She helped out with the translocations, which you'll hear more about today. So this was a collaborative project with many different PIs and graduate students involved. She helped with all aspects of this, including, moving the animals, helping with field work, attaching tags, and helping in the lab. She then used many of her skills to help with the CERDIT project, which is a project studying the impacts of multiple stressors on elephant seals. Again, another collaborative project with you see Santa Cruz, Sonoma State, and U.S. Fish and Wildlife Service. And here you can see her working with another graduate student. And you might think it's kind of silly that she's painting the nails of an elephant seal. She probably did not think that that would be one of the skills, giving manicures to seals. But this actually really helps us relocate the animal, because you can see they have the marks on them, but they molt all of their fur off. So having these nails that don't molt off colored helps us find the animals we're interested in later. So she became a really important team member of that. And then back at Moss Landing, she was very important for our stranding program. She kind of handled the data management for a couple of years, and her and Lauren worked quite closely. You can see there, they were out on a large whale response. She was an essential lab member for many different reasons. She was always a great participant in lab meeting, offered good suggestions, but she also would help the students out in the field. Here you can see her and Sierra, where she's out helping Sierra with her sea otter observations. She would always be willing to step in for things like bowling, and was just a good friend to a lot of the team members or the lab members. And then she made good use of her time also scientifically. So she participated in 2 different conferences in her time where she presented some of her thesis work. The first was virtual biologging conference, and then she went to the Marine Mammal Conference in 2022. She also received several awards and scholarships, including the CSU Coast Graduate Research Award, the Myers Oceanographic and Marine Biology Trust, the Moss Landing Scholar Award 2 different times, and a Moss Landing Wave Award. And with that, I will pass it off to Daphne so she can tell you more about what she's been working on the last few years.
All right, thank you so much, Geeta, for that wonderful introduction, and thank you everyone for coming to my thesis defense. I will be talking about the behavioral response of diving northern elephant seals, Mirounga angustirostris, when exposed to predator calls. So before we get started, I just want to mention that there will be a progress bar on the right-hand side of all of the slides. So throughout the presentation, you can look over there to see which section we are in. With that, let's get started. The Snow of Fisheries infographic depicts the importance of sound in an underwater environment. Noise in the ocean has increased dramatically, especially due to anthropogenic sounds from ships, construction, sonar, seismic surveys, and more. Marine animals use sound and hearing for a variety of daily tasks, such as foraging, navigation, communication with conspecifics, and detection of nearby predators. Since sound travels fast and far in water, excess noise can disrupt marine animals' ability to complete these important tasks. Therefore, it is critical to understand how acoustic stressors impact the behavior of animals in the ocean. This figure is from a paper that reviewed marine mammal responses to noise and broke it up into 3 categories: behavioral, acoustic, and physiological. And responses can range from subtle to extreme and depend on the animal's physiological capabilities and its behavioral state prior to exposure. Studies that examined stress typically looked at physiological responses, such as hormone levels, heart rate, or oxygen usage. Others have looked at how acoustic exposures can interfere with an individual's ability to detect sounds for communication or predator awareness, or cause an animal to change its vocalizations. Then there's behavioral responses, which include any changes in locomotion of an individual. And I specifically want to look into these behavioral responses. So both short-term and long-term effects can be seen with behavioral responses, which have been examined in various whale species. Whales have been observed immediately swimming away from projected sound sources, diving deeper, diving longer, changing surface intervals, altering activity metrics, or decreasing or stopping feeding behaviors when exposed to acoustic stressors. And if an individual reduces foraging time or spends excess energy avoiding stressors, it may lead to long-term effects, including decreased fitness or reproduction ability. And in extreme cases, it could end in death, as is suspected with several species of beaked whales that were thought to have mass stranded due to decompression sickness as a result of being exposed to naval sonar. The type of acoustic stimuli can also impact the behavioral response of marine mammals. Some studies use natural sounds, such as those from conspecifics or predators. The killer whale is one of the top predators of many marine mammals and often causes an escape response from their prey. Some other studies use anthropogenic sounds, such as ships, drilling, and sonar. And coincidentally, killer whale calls sound similar to a type of naval sonar called mid-frequency active sonar. And these two sounds have caused comparable reactions in various study species. If animals consider sonar to be an equivalent threat as a predator call, causing them to perform evasive behaviors, then an increase in anthropogenic sounds in the oceans may lead to negative effects on marine mammals. This figure comes from a study that examined how 4 cetacean species responded to acoustic stressors. I know there's a lot going on, but I want you to focus on these red rectangular boxes. The boxes represent the time when exposures were happening, and you can see that the behavior recorded inside those boxes looks different than the baseline from the previous few hours on the left side of each plot. The researchers found that the species that stopped foraging more quickly when hearing killer whales also decreased foraging when hearing sonar, which shows that species with strong anti-predator responses also respond strongly to anthropogenic noise. The variation between previous studies creates challenges when comparing behavioral responses, mainly due to differences in received sound levels and methods for observing behavior. Playback experiments often broadcast acoustic stimuli from nearby boats or transmitters on the seafloor. Then the level of exposure is measured by a hydrophone on the animal or by estimating the distance between the target animal and the sound source. Also, studies either track specific individuals using motion sensor tags to examine underwater response, or they documented responses of all nearby animals using visual observations from the surface. Aspects such as location of sound source, distance, familiarity, and speed of the sound can impact behavioral responses. Since many researchers do not know the behavioral state of their target animals until tags are retrieved, assessing responses to repeated exposures under consistent conditions has been challenging. To better control aspects of acoustic stimuli, such as timing of exposures, received sound level, and repeated exposures under consistent conditions, a new biologger was developed that combines an experimental acoustic playback with a passive acoustic recording system and a three-dimensional movement recorder. This tag can be programmed to play an acoustic stimuli during specific portions of the dive cycle, which allows for consistent repeated exposures paired with behavioral observations. A preliminary study proved that the biologger could elicit responses in tagged free-ranging individuals, as seen in the bottom left photo. And now this tag design on the right is made commercially by loggerhead instruments. Northern elephant seals are an ideal study species to test the behavioral response of deep diving marine mammals to acoustic stressors. They have great underwater hearing sensitivity. Their dive behavior is well studied, which allows us to identify how a noise disturbance modifies their behavior. They are accessible to work with since they breed and molt on land. They handle instrumentation well, meaning they respond well to the procedures and behave similarly with or without a tag. And they have a homing behavior that allows researchers to retrieve fine-scale data recorded on the tags. To answer my question of what kinds of behavioral responses diving northern elephant seals have when exposed to acoustic stressors, I had the following 4 objectives. 1, compare dive characteristics of exposure dives versus non-exposure dives. 2, compare fine-scale behavior before and after dive inversions that occurred during exposure dives with playback on ascent. And I'm specifically interested in comparisons that are at a much finer scale than previously done. 3, measure how long it takes to return to normal diving behavior after acoustic disturbances. This has previously been a challenge due to difficulty following individuals for extended periods, but the new tag allows us to learn about return to normal dive patterns and other fine-scale behaviors. And 4, determine how seals respond to repeated exposures of predator calls, and looking at if they show any signs of habituation or sensitization to the chronic exposures. Survival instincts should discourage an animal to completely habituate to predator calls over time, but it is also costly to constantly be exerting extra energy. All right, now let's get into how all of this was done. Before I go into the specifics of my project, I want to acknowledge that my thesis was just one part of a larger NSF-funded project titled At-Sea Experimental Disturbances to Characterize Physiological Plasticity in Diving Northern Elephant Seals, with the PIs listed there. Also, since we were working with wild elephant seals, all activities I'll be describing were conducted under these National Marine Fisheries Service permits, as well as appropriate IACUCs. To start off, we were able to do this experiment with the use of translocations. The translocation paradigm with elephant seals is based on the fact that they have a homing behavior to their natal beach. So if an animal is dropped off somewhere, it will want to swim back home. I'm going to go over an overview of the field portion of the project and then go more in depth in the next few slides. So first, we pick up an animal from Año Nuevo and bring it to the lab for instrumentation. Then we bring it down to Pacific Grove and drop it off at a beach. And if all goes well, the seal will swim back to Año within the next week or so. There were 3 field seasons for this project in 2018, 2019, and 2021, during which we instrumented a total of 22 northern elephant seals. Our fieldwork was conducted in March and April because we wanted to work with the seals before their annual molt. We would go out to Año Nuevo and choose a healthy unmolted juvenile seal, so 1 to 2 years old. And we want to pick an unmolted animal, so there's old fur to attach instruments to and ensure that those animals will want to return back to land with our biologgers. Once we choose the seal, we chemically immobilize it using an intramuscular injection of teolazol. And once the seal was sedated, we took some blood samples and rolled it into a metal cage, as seen in the right-hand side of that photo. Then we loaded the cage onto a truck and transported it to Long Marine Lab at UC Santa Cruz. Upon arrival, the seals were weighed in the cage and moved to an animal holding area to rest until the instrumentation procedure began. Following the recovery period, the seals were again immobilized with an IM injection. Then sedation was maintained with isoflurane gas anesthesia using mask induction, as seen in the photo, followed by intubation. Our instrumentation procedures were about 2 to 3 hours long, so this method worked best for the seals. We used epoxy to glue the following biologgers onto the seal's fur. The tags were wrapped in splicing tape and attached to a mesh, as seen in the photos, to allow for easy removal later. The AMX tag or acoustic tag was the one collecting all the data that I'll be talking about, and it was attached off-center near the shoulder of the seal. The satellite tag was attached to the head to allow for the best transmission of location data via the Argo system to track the seal's movement back to Año. The VHF tag would allow us to pinpoint the animal's location after it returned to land. And 2 additional tags, the daily diary and the oxylog physiological logger, were attached for use in other studies. After the seals recovered from anesthesia, they were loaded onto the truck and driven down to Pacific Grove. We released the seals on the beach and they began their journey back to the colony, collecting valuable sound and movement data while they dove deeply across Monterey Canyon. When the satellite tags showed they were back on land, our team went out to find our seals again. We would use a VHF scanner to listen for our seals' tags to find them in the colony. And they were immobilized again and biologgers were removed. Then the remaining mesh and epoxy patches would molt off when the seal molted over the next few weeks. The AMX tag was designed to record passive acoustics, pressure, temperature, light, and 3-axis motion using a triaxial accelerometer, magnetometer, and gyroscope. Playbacks of a preloaded sound file produce the sound exposures heard by the seals, which in our case was a 30-second recording of a transient killer whale whistles. And here's hopefully a short clip of that sound. The tag used a time depth sensor to trigger playbacks at program time intervals and dive stages. It was meant to play the exposure a maximum of 25 times during the translocation with at least 2 hours between playbacks. It was only supposed to be triggered when the seals reached 125 meters above their maximum dive depth after descending deeper than 250 meters. But the tags occasionally had some malfunctions and accidentally played the sound during some descent phases as well. Like I said earlier, all the data I used was collected from the acoustic tag. First, I had to convert those files into usable files for data processing. Then I audited all the sound files recorded by the tag's hydrophone during the translocation. I used a program called Raven, which allowed me to both listen and visually inspect the recorded sounds in spectrogram form, as seen in that image. So much data was recorded for each seal since some sensors were recording at 100 Hertz for up to about 4 days. So everything was stored in 1-hour segments that I combined into a single file for each seal. Then I created sequential timestamps for the files so that even though they were recording at different frequencies, data from all the sensors would line up properly. For 2-dimensional movement processing, this is referring to what I got from the pressure data, which was converted into depth to create a TDR record or a time depth record, and then plotted as a dive profile. Whenever I show dives, you'll be looking at depth on the y-axis and time on the x-axis, and this is showing a complete translocation for one of our seals. I then used the IGNOS toolbox in MATLAB to analyze the data. This figure on the right shows an example output with what IGNOS labeled as dives based on my thresholds, and it's the same sequence of dives as in that red box. Depth data was 0 offset corrected to account for pressure sensor drift and ensure surface events were accurate. Basically, this fixes tag recording errors, so the seal is at sea level when it's surfacing and not flying 5 meters above the ocean surface. Then I selected thresholds consisting of a minimum dive depth of 15 meters, a minimum dive duration of 30 seconds, and a fixed bottom phase of 80% max depth. The IGNOS program calculated dive parameters, and I used the following ones for my analysis, including the dive number and the date and time for the start of the dives. And this figure is showing a single dive. Maximum depth was important because I only looked at dives deeper than 250 meters, since that was one of the parameters for the AMX tag for playing exposures. Dive duration consisted of a time a dive started to when the seal was back at the surface. Descent rate and ascent rate were found for any of those times outside the bottom phase of a dive, so above the 80% max depth. And post-dive interval is the amount of time at the surface between dives. To separate the acoustic exposure dives from the non-exposure dives, I used the timing of playbacks found when I audited the sound files. The non-exposure dives on the right were V-shaped. Then the exposure dives were further broken up into dives with playback on descent in the middle and playbacks on ascent on the left. Then the exposure dives with playback on ascent always had a dive inversion or a reversal of dive direction, as seen from that W shape. The dive inversion separated the two halves of the dive into pre-inversion and post-inversion sections, which I later used for fine scale analysis. Next, I work with all the three-dimensional movement data to calculate 4 dive variables, which I will go into further detail in the next few slides. The accelerometer, magnetometer, and gyroscope all collected data in 3 dimensions. This picture helps show the types of movement that an accelerometer picks up, and it's just like what's inside your smartphone or smartwatch to count your steps or know if you tilted your screen. To work with the fine scale variables, I downsampled the data since the behavior I'm looking at didn't need to be as exact as 100 data points per second. And also I want to mention that I use the tag tools package in R for many of these calculations. All right, now let's start the 3D data processing with stroke rate. Flipper strokes were calculated using a custom written code in R. Elephant seals stroke side to side with their rear flippers, and that kind of motion is most visible on the y-axis accelerometer data. So I started by running that through Butterworth filter. This extracted the dynamic acceleration and centered the data, which is seen in the before and after on the bottom of the slide. Next, I used a peak detector with set criteria. The figure on the right is a 1-minute chunk of the data on the left. Random trials were conducted for each seal to check the accuracy of the detector against my manual count. I adjusted the threshold where the red dotted line is until the timing of the peaks where each of the yellow dots are was consistent with my manual count, and the dots represented each flipper stroke. Next, I calculated ODBA. Overall dynamic body acceleration is a common acceleration metric that gives insight into how much movement or energy an animal's using. There are several ways to calculate ODBA, and I chose the method that used the accelerometer data to calculate the seal's dominant stroke frequency, then sent the data through an FIR filter. The output is a single integrated measure of body motion in 3 dimensions. I've included a figure showing a single dive on the top and the corresponding ODBA on the bottom as a visual. And you can see that where that dive inversion occurs in the middle of the top image, the bottom image is different right at that point as well. Then I calculated pitch, which gives the angles of rotation around the y-axis. Basically, I'm looking at if the seal is facing up or down. And what you can see in this figure is that there is a change right at the time when the dive inversion occurs again, which was why I wanted to look at how pitch differed during different sections of exposure dives. Finally, I calculated heading of the seals, which was the direction they were facing in relation to magnetic north. Tag Tools does the calculation using both the triaxial accelerometer and magnetometer data, and I converted the results into degrees. And again, you can see that there's quite a bit of variation in heading and a clear change right at the time of that dive inversion. After the 4 fine scale variables were calculated, I needed to split them into 4 categories to be compared. The pre-inversion and post-inversion sections were separated by the timestamp of the start of the acoustic stressor playback, as seen by the killer whale noise symbols. So picture it as the first V and second V of a W-shaped dive. Then each of these V's had an ascent and descent section, and together this gave me the 4 categories. Then the 4 categories were each made up of 60-second segments. Stroke rate was calculated by counting the number of strokes in each section and dividing by 60. For ODBA, I found the mean of the acceleration metric within that 60-second segment. And for pitch and heading, I found the range of the variables within each segment by subtracting the minimum from the maximum. And this provided the greatest amount of change in pitch or heading observed during each 60-second segment. Now to get into the stats, my first objective was to compare dive characteristics of exposure dives versus non-exposure dives to determine if seals exhibited behavioral changes in response to the acoustic stressor. I used linear mixed effects models to assess differences between each of the 2-dimensional dive parameters listed there: dive duration, descent rate, ascent rate, and post-dive interval, during the 3 dive types, which were playback on ascent, playback on descent, and non-exposure. And I'll be showing these images again later as a reminder of which objective I'm talking about. My next objective was to compare fine scale behavior before and after the dive inversions that occurred during exposure dives with playback on ascent. I again used linear mixed effects models to examine differences between each of the 3D dive variables, which were stroke rate, ODBA, pitch, and heading, during the 2 dive portions, pre-inversion and post-inversion. So basically, I compared the dark purple to dark green and light purple to light green. My third objective was to measure how long it takes to return to normal diving behavior after acoustic disturbances. I used linear mixed effects models again to investigate when seals return to baseline following an exposure dive for each of the 4 whole dive variables: dive duration, descent rate, ascent rate, and post-dive interval. Baseline was determined using the 2 dives prior to the exposure dive, which are shown in red. Then there's the exposure dive in green, the first dive after exposure in blue, and the second dive after exposure in purple. I'm trying to see how many dives it takes to return to normal, or if it's still not back to baseline by that time. Since elephant seals dive for such long durations, and we set our acoustic tag to play the sound at 2-hour intervals, I couldn't look at dives further than 2 post-exposure dives. My final objective was to determine how seals respond to repeated exposures of the predator call, like if they showed signs of habituation or sensitization to the killer whale whistles over time. I used linear mixed effects models to learn if after multiple exposures, do those variables change? And I did this analysis in 3 sections: the whole dive level, 60-second fine scale descent, and 60-second fine scale ascent. Also, for these fine scale variables, I looked at how much variation there was between pre-inversion and post-inversion values. Since I was looking at the effect of repeat exposures, I only included the seals that were exposed to the at least 5 playbacks of the killer whale whistles. So the seal whose dive profile is shown here was removed from analysis since it was only exposed 4 times during the translocation. All right, now let's get into what I found. First, I want to start off with some general results. Of the 22 acoustic tag deployments we did, 18 of them were successful, and they were spread throughout those 3 field seasons. Unfortunately, the tag failed to record data during the other 4 deployments. The acoustic tag collected audio and motion data for 1.5 to 3.8 days before it either ran out of battery or the seal returned to the beach. Our seals spent between 1.5 and 48.5 days at sea during their translocation, with a median of about 3.5 days. During that time, the elephant seals were exposed to the killer whale whistles between 1 and 25 times, depending on how long they were at sea and if they were consistently diving deep enough to trigger those playbacks. There were a total of 211 exposure dives among the 18 seals, and of those, 153 happened on ascent as programmed, and 58 happened on descent due to some tag malfunctions. Every time the seals were exposed to the killer whale whistles on ascent, they performed a dive inversion, and when they were exposed on descent, they tended to swim faster. Also, there were 3,613 non-exposure dives found using the parameters I chose, and I ended up using 705 of those for analysis because they were deeper than 250 meters, which was the minimum threshold for acoustic exposures. Again, my first objective was to compare dive characteristics of exposure dives versus non-exposure dives. These are the same figures as earlier. Now they're going to be moving to the top right of the screen. In this figure, the colored box plots represent the 3 groups of dive types, with dark blue representing exposure dives with playback on ascent, light blue are exposure dives with playback on descent, and pink are non-exposure dives. And this is the color scheme for the next few slides as well. I found that there were differences in dive duration between the dive types, with exposure dives with playback on ascent having the longest average dive durations at about 29 minutes. Then exposure dives with playback on descent were slightly shorter at about 27.5 minutes, and non-exposure dives were the shortest, averaging about 22 minutes. You can see on the box plot that there's a pretty big difference between the non-exposure dives and exposures on ascent. During those, the elephant seals were consistently performing dive inversions that greatly extended the length of their dives. For descent rate, I also found differences between the 3 groups. Dives when the sound played on ascent had slower rates than both non-exposure dives and exposures on descent, which was a bit strange. Since this was unexpected, I looked at the data a different way, and when I grouped these differently and combined descent rates of all dives with no playback on descent, so those pink non-exposure and the dark blue exposure, then descent rate of the light blue playback on descent was faster than the other 2, which made a lot more sense. I think my sample size and the range of values we're seeing help explain that. There was also a difference in ascent rate between the 3 different types of dives. Dives when the sound played on ascent had a faster ascent rate than the other groups, and ascent rate for non-exposure dives and exposure dives with playback on descent did not differ. Finally, there was no difference in post-dive interval between the 3 dive types. My second objective was to compare fine scale behavior before and after the dive inversions that occurred during exposure dives with playback on ascent. As a reminder, I'm comparing the dark purple to dark green and light purple to light green. Looking at the descent portions of exposure dives, stroke rate, mean of ADVA, range in pitch, and range in heading were all greater during the post-inversion section, which is shown in green. This means that after the seals completed the first V of their dives and started going back to the surface, they heard the killer whale whistles, performed a dive inversion, and started stroking faster, stroking more energetically, had greater changes in pitch, and had greater changes in heading. For the ascent portions of the exposure dive, stroke rate, average ADVA, range in pitch, and range in heading were also all greater during the 60-second post-inversion section, again shown in green. This means that after the dive inversion, as the seals turned around and started swimming back towards the surface again, they were still stroking faster, stroking more energetically, and had greater changes in the angle of ascent and their swimming direction compared to the initial ascent from the first portion of their dive. My third objective was to measure how long it takes to return to normal diving behavior after acoustic disturbances. And for this, I was comparing the blue first post-exposure dive and purple second post-exposure dive to red baseline dives. In the top left, dive duration of exposure dives was longer than non-exposure dives, both before and after, and elephant seals returned to baseline dive durations immediately following exposure dive. In the top right, descent rate of the second post-exposure dives in purple were slightly faster than the baseline dives in red, but all other combinations were the same. In the bottom left, ascent rate was faster during exposure dives compared to all non-exposure dives, and the post-exposure dives had slower ascent rate than the baseline dives. And in the bottom right, post-dive interval was slightly longer after exposure dives, but it quickly returned to normal on the next surface interval. My fourth objective was to determine how seals respond to repeated exposures of the predator call. And before I go into the results of this objective, I want to mention that all elephant seals continue to perform dive inversions when exposed to the playback on ascent, no matter the number of repeated exposures. So overall, the seals had strong behavioral response to the acoustic stimuli each time and did not fully habituate and start ignoring the sound, which is critical for their survival. But I did see some small behavioral changes over time. First, these are the 4 whole dive variables examined. As the elephant seals experience an increasing number of playbacks, their dive duration, as shown in the top left, during exposure dives decreased. And during exposure 1, the seals averaged a dive duration of 34.7 minutes, but by exposure 10, that decreased to 29.5 minutes. And by exposure 25, the average dive duration dropped to 23.1 minutes. In the top right, you can see that there was no trend in descent rate as seals were exposed to more playbacks. For ascent rate in the bottom left, seals appeared to increase their ascent rate as they heard repeated exposures. During the first exposure, seals had an average ascent rate of 0.63 meters per second, and by exposure 25, the average ascent rate went up to 1.03 meters per second. And you can see that there was a lot more variability in the earlier exposures compared to the later ones. Finally, in the bottom right, post-dive interval remained steady as seals were exposed to the acoustic stressor repeatedly. Also, when I examined these dive variables in relation to increasing numbers of non-exposure dives, there were no trends observed, meaning that these results are due to repeated exposures and not the amount of times the seals spent at sea. When examining acoustic disturbance at a fine scale during descent sections of exposure dives, there were no trends observed in any of the variables. Stroke rate, ADVA, pitch, and heading all did not differ between the pre-inversion and post-inversion descents, meaning that at a fine scale, the elephant seals responded the same no matter the number of exposures. The variation of these 4 fine scale variables also did not change between the pre-inversion and post-inversion ascent sections of exposure dives after repeated exposures. So no matter how many times the seals heard the acoustic stressor, they still responded the same during the 60-second ascent sections I looked at. Right, and we can discuss a bit more and talk about what the results might mean. The juvenile northern elephant seals exhibited strong behavioral responses to the killer whale whistles. The most distinctive was performing dive inversions following each playback on ascent. These dive inversions caused extended dive durations, with exposure dives averaging about 29 minutes compared to deep non-exposure dives averaging about 22 minutes. And this follows trends seen in several species of whales that also dove deeper and extended dive durations when exposed to acoustic stressors. I thought that post-dive interval would increase after exposure dives, but that wasn't the case. The one anomaly we had was Candle, who completed a 49-day migration rather than the normal 2 to 10-day transit. But Candle's post-dive interval behavior more closely matched that of adult female northern elephant seals that occasionally perform extended surface intervals during their foraging migrations. Unfortunately, the tag's battery only lasted a few days, so I don't know what his behavior was past that first week or so. Breath hold divers use surface intervals to offload carbon dioxide and replenish oxygen stores, which was why I thought our animals would extend their post-dive interval. But the species is known for performing long, deep dives without extending surface intervals. So most likely the extended dives did not completely deplete oxygen stores to a level that required longer recovery times. Also, elephant seals likely feel safer at depth since they can dive deeper than their top predators, which are primarily surface hunters. And therefore, dive inversions, continuous diving, and minimum time spent at the surface likely lead to the best chance at survival. Since I believe that the extended dive durations after unexpected exposures to killer whales did not completely deplete oxygen stores, I wanted to talk about what we know about elephant seal dive physiology to support this idea. Elephant seals must have some physiological plasticity that allows them to conserve oxygen. During normal dives, they have a drastic decline in heart rate at the start of the dive, then an anticipatory tachycardia as they start to ascend, which becomes more pronounced in the final 15 seconds before surfacing, as shown in this figure from Andrews et al. And blood oxygen typically declines throughout a dive, but drops more rapidly during the final 15 to 45 seconds of ascent, as shown in this figure from Mir et al. And also, it's been found that elephant seals don't always fully replenish their oxygen stores during post-dive intervals. Since the seals in my study were exposed to the killer whale whistles early in the ascent phase, likely before they sharply increase heart rate, which depletes oxygen stores. So at the time of the acoustic disturbance, the seals are probably still able to ration their oxygen to extend their dive. And I'm interested to see what was recorded by the seals' physiological loggers and combining those results with these behaviors. Now I want to look a little closer into some of the exposure dives. When the playback happened on ascent, the seals had faster ascent rates. It's been observed that some large whales, when they're exposed to acoustic stressors, return slowly to the surface, while others execute high-powered ascents. So elephant seals seem to fall into that second category. This will allow them to quickly offload carbon dioxide, reload oxygen, and spend less time in shallow waters where predators could be hunting. Also, even though the seals are likely capable of longer dives, it might be beneficial to return to the surface quicker before another disturbance causes the need for an additional dive inversion. When playbacks happened on descent, whole dive variables were almost the same as during non-exposure dives. Most likely, since the playback happened early on in the dive, the seals recovered from the disturbance very quickly and therefore didn't show many differences in dive variables. Decompression sickness is a concern for some whale species, but for the elephant seals, it appears that they can adjust their diving in a way that does not have the same negative effect for their health. On a fine scale, the seals increase their activity and variation in orientation, just as I predicted. The increased activity metrics and steeper pitch were likely from the seals trying to escape the predator call, which was similar to what has been observed in whales that also increase their swim speed, stroke rate, or ADVA in response to acoustic stressors. One cool thing I found was that many of the seals appeared to spin in circles during the 30 seconds of killer whale whistles, then return to head in the same direction as they were initially swimming. They were likely doing this to try and locate the source of the sound and determine which direction to escape. Other studies have reported whales swimming away from areas with projected sounds, but this spinning behavior has not been reported. This examination of fine-scale movements immediately before and after acoustic exposures isn't really known before because of the past difficulty in knowing exact timing of exposures. So these results now show that seals sustained increased activity, which could increase energy use and lead to decreased resting or foraging time, and therefore overall health. Next, looking at the duration of behavioral responses, I found that seals took action to escape the predator call, but quickly returned to baseline diving behavior immediately following the disturbance. This behavior ensures that extra energy expenditure doesn't continue on subsequent dives when the threat is gone. Even though elephant seals recover quickly when they are transiting, behavioral responses may be different if they were foraging. So with actively feeding cetaceans, it's been found that they'll leave an area for several hours or days. So it would be important to look at how elephant seals react to exposures of acoustic stressors during foraging migrations. The trade-offs between the risk of predation and benefits gained from continuing to forage are very different than when just traveling. Overall, elephant seals seem to adapt well to stressful scenarios, and with anthropogenic sounds in the ocean continuing to increase, the ability to return quickly to normal behavior after disturbances could be extremely beneficial. Finally, I looked at how elephant seals responded to repeated exposures of the killer whale whistles. It seemed like previous exposures could influence the response to later exposures. The seals always perform those dive inversions to escape the sound, but seem to acclimate after repeated exposures, as seen from the decrease in dive duration. Perhaps the seals could more quickly determine that the threat was not present, so they were able to recover more quickly from the fear response. Fine scale behavior remained the same no matter the number of exposures, likely because that escape response is necessary for survival. And habituating to predator calls can be life-threatening, so reduced reactions over time is likely the best solution. To wrap things up, the use of an animal-born biologger that played the predator call under controlled settings allowed for the collection of movement data that is rarely possible. I found that juvenile northern elephant seals exhibited strong behavioral responses at the fine scale and whole dive level, proving they are capable of easily adjusting diving behavior when confronted with an acoustic stressor. The seals recover quickly from the stressful event and immediately return to baseline behavior during the dive immediately following the exposure dive. Finally, even though they respond to each predator call, their behavioral responses are less intense after repeated exposures. And here are just some thoughts on where this research can go next. First, integrating these behavioral responses with physiological measurements will help us fully comprehend how these animals can change their behavior in the way that they do and how these changes impact their health. Future studies should also look at responses when animals start in different behavioral states, such as foraging rather than transiting. More repeated exposures, additional time between exposures, different sounds, and testing on different marine mammal species would also be beneficial. And as sounds in the ocean continuing to increase, it is critical to learn how different sensitive species are being impacted. Right, now if everybody can just bear with me, I have some acknowledgements to get through. There's a lot of people to thank to make this project possible. First, I want to start with my wonderful thesis committee led by Dr. Geeta McDonald. Thank you for accepting me into your lab and being such an amazing advisor. I really appreciate the opportunity to join the translocation project and all the knowledge that you have shared with me, either in the field or the many classes of yours that I took here at Moss. You made me a better writer, and I definitely improved as a scientist because of you, so thank you for everything. To Dr. Holger Klink, thank you for sharing the acoustic tag data with me and your knowledge of the inner workings of the tag. Your help with coding when I was struggling to clean up my messy data was very much appreciated. And to Dr. Scott Hamilton, thank you for your wonderful stats knowledge and your helpful writing feedback. It was great to have you on the committee to remind me to see the bigger picture sometimes when I was too focused on the little things. Next, I need to thank our elephant seal team, which was made up of a lot of people. The larger translocation project was led by 7 amazing PIs, Geeta, Allison, Cass, Paul, Marcus, Holger, and Dan. Thank you all for making this whole thing possible and for letting me carve out my portion of the project. You guys are the best of the best, and I'm so lucky to have had the chance to learn from all of you. And I hope we get a chance to work together again in the future. To the other grad students that were on this project, Sean and Lauren, I'm glad you guys were there to show me the ropes when I joined the project during the 2021 field season. Thanks for passing on everything you learned and for all the fun times, either prepping for the field season or processing our hundreds of blood samples. And to our field team, the people listed contributed so much time and effort into the field work for this project, and there's even more not listed that were part of the project before I joined. So I want to thank all of them as well. Even though our seals were only 1 to 2 years old, they were very heavy, so lots of muscle was needed to lift them. And I'm glad we got to enjoy some early morning sunrises and late nights, and especially in our tent with the twinkly lights when we couldn't go indoors because of COVID restrictions. Next, I want to thank my funding sources for supporting the project through fieldwork, equipment, living expenses, and more. The National Science Foundation funded the larger translocation project, and I received grants from COAST through the Graduate Student Research Award, from Moss Landing Marine Labs through student scholarships, and from the Myers Trust, their award and Baldrige Award. Next, I want to thank the Vertebrate Ecology Lab, the best lab at Moss, in my opinion. Thank you all for your thoughtful feedback on research, but more importantly, the fun times on stranding adventures and whale necropsies and spending time just hanging out outside the lab. Gita, you really picked a great group of people in this lab. Extra thanks to Katie, Parker, and Mason for coding help when I was trying to figure out all of my data. And a special shout out to Sierra, who is my cohort mate in the Vert Lab. Thanks for coming to introduce yourself during Moss Landing orientation our first day. We since then become best friends, and I'm so glad we got to go through this grad school journey together. And some other support that I want to thank. I worked several jobs throughout grad school to support myself, so I needed to thank my supervisors for hiring me. To Dan, Gita, Sarah, and Rachel. Thank you guys for letting me join the CERTUP team. It was such a great opportunity to be able to work with the adult female elephant seals on a project that was similar to my thesis. And I'm so grateful for all the experience I gained and for every second I could spend at Anyo. And a special shout out to Rachel, who not only helped me with the dive analysis for my project, but also taught me pretty much everything I know about elephant seal fieldwork. So thank you, I really appreciate you. To Katie at the MLML library, thank you for being an amazing boss and being so understanding of a grad student juggling a thesis, classes, fieldwork, multiple jobs. And thanks for teaching me all the library things. Because of you, I can now find some of the most obscure journal articles out there. To Karen and Scott, thanks for letting me join the Leatherback project and flying aerial surveys with you guys. It was such a cool experience, and this picture on the bottom right is actually from our survey plane flying above Año Nuevo, so that was a really fun perspective. To MLML staff, especially the people listed there, I want to thank them for help with various grad school paperwork and also rescuing my hard drive when my computer crashed a couple years ago. And to MBO Saturday crew, shortly after I started at Moss, I decided that I want to spend my free time volunteering with marine mammals. So I joined the Marine Mammal Center. And they were such a great community of people to work with. So thanks guys for listening to my random elephant seal facts through the years. Finally, I want to thank my family and friends. My friends near and far have supported and encouraged me through this whole process, and I'm lucky to have all of you in my life. Some extended family, some of which are in the audience today, both in person and on Zoom. Thank you guys for your support. And finally, to my mom and dad, your constant love and support through everything has meant so much. And thank you for always believing in me. I wouldn't be where I am today without you. And with that, thank you all so much for listening, and I'm happy to take any questions.
Questions in the room, if anyone has them, and I'll run it into the queue in case there's somebody on that. Is there like, I guess, any possibility that the anesthetic used had any sort of, problems for the animals in terms of, finding fermentation or swimming or, that kind of thing?
The protocols used have been used previously, so I don't think they really have any negative effects later on. Another person on our team looked at stress physiology and kind of looked at how heart rate and everything changed before, during, and after anesthesia, and found that the elephant seals recovered really quickly. So they're just very robust and a good species to work with.
Thanks, great talk. So you mentioned that they do this consistent diet inversion, which is presumably energetically costly, but that their oxygen concentration doesn't reach, dangerous levels. But given that there's probably like an energy cost associated with it, any thoughts about what that might mean in terms of how much they might need to eat, or what are the energetic knock-on effects that you might consider?
I'm not quite sure if that has exactly been looked at, but I think there's spending excess energy that they weren't expecting to in any way should be costly at some point. So either if that decreases foraging opportunities, if they were actively foraging, or takes away resting opportunities, I think anything like that in the long run could have some kind of effect. But at least in the short term, it's good to know that they recover quickly at least. So hopefully, even though they're responding, it won't have too many negative effects, but this is specifically looking at elephant seals, which are a very robust species. So it's important to look at this also with some other species that maybe are not as good at recovering from stressful events. Sorry, I don't know if that fully answered the question, but.
So you said that you recorded the elephant seals doing maybe circles during the when they were hearing the killer whale calls. Do you think that is because the tag was on them and they actually couldn't really find directionality of the sound?
Yeah, I think so, most likely, just because it's very unnatural for that sound to be just a meter away from their ears. So it's much closer, like in the wild, they'll probably see a killer whale before it gets that close. So the spinning probably is to try and be like, okay, which direction is it in? But with some of the seals, especially, as soon as that sound stopped and they stopped searching direction, it was just like, okay, I can continue in the direction that I was swimming before. But yeah, I definitely think the spinning is because the sound is coming from their back and maybe not further away. If it was coming from a boat, let's say a quarter mile away or something, they would probably just swim in the opposite direction. Yeah.
I was thinking about the habituation, and I'm wondering if they hear a predator calls when they're on a diet and they come back to the surface, are they, I guess, looking for any other cues that there is actually a threat nearby, and that maybe then limit the habituation effect?
Perhaps. Because that post-dive interval after the exposure dives wasn't significantly longer than all the other dives, I think most likely part of the reason is probably because physiologically they don't need that extra time, but also they don't want to be in the surface waters where killer whales and great white sharks could be swimming. So I'm not really sure if they're kind of looking around when they're at the surface to see if any dangers might be coming. But with elephant seals, they know that if they go back to depth, they're most likely going to be safe since their predators can't reach as deep as they can.
Make it easy so far. There's no online questions, but lots of people say great job and congratulations. You read those. All right, well, there aren't any more questions. I think you did a great job explaining.
Thank you.
We can say congratulations. It'll still Daphne away for a little bit and have a committee meeting, I think in my office. There wasn't much room anywhere else in the other today. And then if you guys want to hang out here, we do have some cake, and it looks like some people brought some other snacks too.
There's a lot of food, so please stick around.
And we'll come back with her in about a minute. Or we'll let her come out. And you sent Holger a link, so we will join us.
I did not. I thought you were.
Oh, I thought you were. So, Holger, we'll send you a Zoom link if you're still on, and we'll.
Yes, I am.
Okay, great. So we'll send you a new link and we'll have a quick meeting in my office, just kind of about next steps.
Sounds good. Thanks.
Okay.
Yeah.